Good morning, dear students. So in this session, we will discuss one of the biological method for energy conversion from biomass, that is anaerobic digestion. So anaerobic digestion, we know that it is a biological process in which the microorganisms break down the biodegradable material in the absence of oxygen. So this process must be carried out in the absence of oxygen. That's why it is known as anaerobic. And the primary product, the main product of this is a combustible gas known as biogas. So this is what anaerobic digestion is. The important feedstocks are energy crops, animal waste like chicken waste, beef tallow, and we can also use municipal solid waste and restaurant waste. And the wastewater treatment, the sludge coming from wastewater treatment is another major feed. And certain industrial wastes like sugar industry, paper pulp, all these can be an input to the anaerobic digestion process. And in turn, we will get biogas as the main product. In addition to that, we will get some slurry, which is very good as fertilizer. Some hydrogen also we will get, and in some certain condition we will get some compost also. So all these are the other products of anaerobic digestion, and this method actually was in practice very centuries before. Even in BC, certain literature says that this anaerobic digestion was there. People know that if you are dumping waste in an air-free environment, some combustible gas is coming out. So that type of observation was there even in BC. And what is the component of biogas? The biogas is mainly methane. It contains almost 50 to 75% of methane. Then carbon dioxide, 25 to 50%. The nitrogen is 0 to 10%. So nitrogen will be mostly present if it is an animal waste, because animal contain more protein compared to plant. So in that case, nitrogen will be more. But if it is a plant-based waste, nitrogen will be negligible. Then hydrogen sulfide. So that is an important unwanted gas that is associated with biogas, that is hydrogen sulfide. It will be 0 to 3%. Then some oxygen. So this is a typical composition of biogas. Now we have to understand how this anaerobic digestion takes place with the help of this diagram. So anaerobic digestion mainly have 4 stages. That is, here it is in green color. First stage is hydrolysis. Second stage is acidogenesis. Third stage is acetogenesis. And finally we have methanogenesis. And all these are carried by different types of bacteria. It's not a single type of bacteria that is conducting all these 4 processes. All these 4 processes are carried by different group of bacteria. Now we already studied, we have animal waste as well as plant waste as the feed. So mostly this will be made of 3 types of material, that is carbohydrate, fat and protein. So every plant and animal waste we can categorize into certain amount of carbohydrate, then certain amount of protein will be there, and fat will be there. So the first step is these are complex hydrocarbons with a very long chain, close linked structure. So the bacteria, all other bacteria cannot be easily decompose this complex hydrocarbon. So the first step is hydrolysis. So hydrolysis is carried by either hydrolytic bacteria, or it can be done by enzyme also. We have some enzymes which will increase the speed of hydrolysis. We can use that also. So it is carried by hydrolytic bacteria or enzymes. So what they are doing, they will be break down this complex hydrocarbon into simple hydrocarbons. So this complex long chained cross linked structure is breaked into straight chain simple structures, mainly sugar. The mainly carbohydrate will be converted into sugar, which is glucose, mainly the glucose. Then the protein will be mainly converted into amino acids, and fat will be converted to peptides or fatty acids. So this is our first step. So we are converting the complex molecules, complex carbohydrates, fats and protein into sugar, that is mainly glucose and amino acids and peptides. So after this process, we have another set of bacteria, that is a fermentative bacteria, which carried out acidogenesis. That is, these are converted into short chain fatty acids like butyrate, pyruvate, propionate. So these are some acids, organic acids, short chain fatty acids. So the second step, these are converted to these acid forms, short chain acid forms. So the pH at the time of this process must be around 5 to 7, that is in the acidic range, or neutral also it can carry it out. So that is our second step. And the third step, the acetogenesis bacteria. Acetogenesis bacteria convert these fatty acids to acetic acid or acetate. So that is our third step. So acetogenic bacteria changes this fatty acid into either acetic acid or acetate. And finally, we have another methanogenesis bacteria, which is very important. So this methanogenesis bacteria, what they will do, they will convert this acetate or acetic acid or carbon dioxide, etc., into methane. So main action of methanogenesis bacteria is to convert this acetic acid into methane plus some carbon dioxide. And this carbon dioxide again will be converted to methane. If there are more hydrogen is present, these bacteria combine this carbon dioxide in hydrogen to methane. Now, what are the factors affecting biogas yield? The first type of digester, the first factor that affects the biogas yield is which type of digester you are using. Then another factor is the percentage of solid content in the waste, that is the percentage of dry matter in the waste. Then another important factor is the C/N ratio, that is the carbon to nitrogen ratio in the feed. Then temperature also affects. Already we have discussed the temperature range at which the different bacteria works. Then pH of the solution is another important factor, since the acidogenesis and the methanogenesis are the rate of these reactions depend upon the pH of the solution. Then retention time, how much time our biomass is present in that reactor. And a type of microbes. So these are the factors affecting biogas yield. And this is a typical flow sheet of anaerobic digestion. So first we have to prepare the biomass before anaerobic digestion, that is shredding. That is, if it is our biomass is of large size, for example our food waste etc. of large size, it should be minimized. And blending, we have to blend the food waste or any other waste so that more or less uniform composition is obtained everywhere. Then pH adjustment, if the pH is not in that optimum range, we have to adjust the pH with adding some acids or other solutions. Then after this pre-treatment of feed, it is fed to the anaerobic digester. So anaerobic digester will be mesophilic or thermophilic depending upon the bacteria. Then you will get mainly two output. One is the biogas, which is a mixture of methane, carbon, hydrogen sulfide, etc. So after that you have to separate these gas for different application if required. On the same time you will get an effluent or a solution which is a biofertilizer. So you can either directly use it or you can remove the biosludge and use for other purpose. So this is a typical flow sheet for anaerobic digestion. Now we will discuss the different types of biogas digesters. So already we know what is biogas digester. It is a closed container in which the segregation and feeding of organic substrate takes place. And in this digester, the biodegradation of substrate also takes place under anaerobic condition in the presence of bacteria producing a methane-rich biogas. The digested slurry can be used as a fertilizer or soil conditioner depending upon its characteristics. So mainly we have three classifications for biogas digesters. Broadly we can classify into three categories. So one is the fixed dome type biogas digester. This is also known as the Chinese type biogas digester because it is firstly in use in China. Then we have a floating drum type biogas digester. This is an Indian type known as Indian type digester because it's come into use mainly in India. And we have finally a bag type digester which is also known as Taiwanese type digester. So this is the three classification of biogas digester: fixed dome, floating dome and bag type biogas digester. Then other than this we can classify a few other classification or categories also. For example, on based of feeding type we can say continuous and batch type biogas digesters are there. Continuous means we can continuously add material and the biogas production will be also continuous. But mostly we will have a batch type digesters where the material is loaded in a one single time and it is left for some period of time and we will get the biogas. So it is the batch type reactors. Now we will see what is fixed dome type digesters. So as mentioned, this was found in China, Jiangsu, China when in 1936. So biogas digesters were in use a few hundred years ago itself. And this is 1936 in China. This was originated, this concept of fixed dome type digesters. And this is a schematic of fixed dome. You can see a reactor with a dome and we have an inlet pipe for feeding this biomass waste and we have outlet pipe for slurry. And the reactor, we have at the top of the reactor we have a gas pipe also. So it consists of an airtight containers. This is the reactor itself and it is made of brick, stone or concrete like material. And we have the feed pipe and digestate pipe, the fixed dome, and at the top of fixed dome is used for gas storage. So the reactor itself, the top of the reactor or digester itself is used for gas storage, and the reaction and biogas storage chambers are connected. So we don't have any separate chamber for this anaerobic reaction and gas storage. We have a single reactor where both will happen. At the bottom we will have reactions and slurry, etc. And the top we have gas because it is having less density. The gas will be always present at the top of the dome. And the sealing is achieved by building several layers of mortar in the inner surface. So sealing is very very important. If sealing is not perfect, means there will be a gas leakage. And in India, this model was first, the fixed dome design in India is named as the Janata model by Gobar Gas Research Station Ajitwal in 1978. So that is the Indian version of this, first Indian version commercially available in this fixed dome category, Janata model. This is a typical figure of that Janata model. You can see some slight difference are there. So instead of pipes, inlet and outlet, we have some cylinder like structures, and some difference is there. The main feature of this model is the digester and the gas holder are integrated parts of a brick masonry structure. So it is made of a brick. The complete structure is made of a brick masonry. And the digester is made of a shallow well having a dome shaped roof. So in the middle we can see the digester, which is having a, it's a well, it's actually a well shaped, a shallow well shaped with a dome shaped upper part. And we have two tanks, the inlet and outlet tanks, connected with the digester through a large chutes, which are called the displacement chamber. So these chambers are known as displacement chamber. On the left side we have an inlet chamber where our feed is happening, and the right side we have an outlet chamber where slurry will be taken out. And the gas pipe is fitted at the crown of the masonry dome, and there is an opening on the outlet wall, on the outlet displacement chamber for the digester slurry. So the digester slurry is taken out through the right side outlet chamber, while the gas will be taken out through the gas pipe at the top of the dome. But the problem with this design was we have several constructional difficulty and also the gas leakage. So these were the two problems faced by this design. Mainly we have construction difficulty when making these chambers and these passages, etc. And another problem was the gas leakage. So due to this reactor suddenly become obsolete, and it is replaced by a new type of fixed dome digester, that is Deenbandhu model. This is very famous model, even in use now also. So Deenbandhu model was biogas plant developed by Action for Food Production, AFPRO, New Delhi in 1984. So that is the most recent type of fixed dome digester available, that is Deenbandhu model. And you can see the construction etc. is simplified. We have some advantage for this model, and the basic principle behind this plant was to minimize the surface area, so that we can reduce the installation cost without sacrificing the efficiency of the plant. So you can see we have a spherical dome like structure, and the complete digester you can see in a spherical shape, hemispherical shape, and the bottom also we have some spherical structure. So that means we have a minimum area in the same volume, so that we can reduce the installment cost and other construction difficulties. And as told, this design consists of two spheres, one on the other. One is the bottom sphere, you can see where slurry will be stored, and at the top we have another sphere. So these are joined, the different diameter spheres are joined at the base, and it is also having an inlet and outlet connected with an inlet pipe and outlet tank. So here it is simplified, the design. The Deenbandhu design is more simplified version of a Janata model. There is no inlet chamber. Instead we have only an inlet pipe coming from a mixing tank, but outlet chamber is present where the slurry is stored. The upper part above the normal slurry level of the outlet tank is designed to accommodate the slurry to be displaced out of the digester. So if a particular design level is reached, the slurry goes out of the reactor to the outlet chamber, and where there is accumulated in the outlet chamber and can be taken out through the slurry outlet. So that is the principle behind this. So we have minimum area, simplified design. Instead of inlet chamber, we have an inlet pipe only, and initially the slurry is stored at the bottom of the digester, but once the level is increases, it moves to the outlet chamber, and after reaching a particular level it can be taken out. So that is this fixed dome Deenbandhu model design. And the advantage, we have several advantages about this model. Mainly the entire digester is in the underground, so it provides a good insulation. So the sealing problem, the gas leakage problems, etc. are very less, because the entire this digester is below the ground. Only we have the mixing tank and the top of the outlet chamber above the ground. All other parts are come under the ground, so it is having a good insulation and gas leakage, and therefore since it is in underground, it have an improved stability due to the variation in temperature. So already we have told the atmospheric temperature conditions will affect the working of an anaerobic digester, but here since the digester is in the underground, that variation will not affect that much because it is insulated from the atmosphere, and it is a more crack proof design. The leakage is very less, and already we have told the minimum surface area is having, so it's have less building materials and installment costs, etc. will be less. And another advantage is, if you have some doubt in gas leakage, even one person can go inside this reactor through that outlet chamber. You can see the outlet chamber is very big, so a person can enter into that outlet chamber, and through that hole he can enter into the digester also. So very easily we can check the leakage in this design. So these are the advantages of the Deenbandhu model. Now overall, what are the advantages and disadvantages of fixed dome type digesters? Mainly they are compared to other two, that is a floating type and bag type digester. The advantages of fixed dome digesters are, first one is it is having low initial cost. Then the lifespan also is very high. Mostly we will get 20 years or greater than that lifespan. Then constructional cost is also very less, and the space also in every model get reduced, and especially in the Deenbandhu model the space is very less. So these are the advantages of fixed dome type digesters. But the main problem or disadvantage is we require highly construction technical skill for a gas tight construction. So this is made of mostly brick or mortar, cement, etc. So it should be highly gas tight construction, otherwise there will always be a problem of gas leakage. And the special sealant, these gas holders also require special sealants also. So these are the few disadvantages of fixed dome type digesters. And another important thing, we have fluctuating gas pressure. The gas pressure is not constant here, since it is not a flexible dome. The dome is stationary. The pressure exerted by the gas will be always varies. So the outlet gas we are getting will have a varying pressure. And already we know the excavation can be difficult in rock, bedrock, or if our location is having a lot of rock underground, it is not possible to install this type of reactors because most of the parts are in the underground. So it is not suitable when there is a lot of bedrock structures in the ground. These are the overall advantages and disadvantages of fixed dome type digesters. So you can see the few figures of fixed dome type digesters. First one is a fixed dome type digesters installed in India. The other one is in Kenya. So mostly these digesters are in use in developing countries like African countries, China and India, because one is due to their ability to convert waste into energy. So all these developing countries lacks a lot of energy in rural areas, and also waste generation is another problem. And compared to other bioenergy techniques like gasification, combustion, etc., these are very cheap also, and this can be employed decentralizedly. So that is why mostly they are widely seen in India, China and African countries. Now we are coming to the second type of digesters, that is floating drum type digesters. So this is involved first in coming in India, not like other one where it is coming in China. So Jashubai J. Patel from India in the year 1956 has first designed the first floating drum type biogas digester, and there hence it was commonly known as the Gobar gas plant at that time. So this is a typical floating chamber reactor. What is the main difference here? Here also we have an inlet pipe for feeding the biomass. Here also we have an outlet pipe or outlet pit for removing the slurry, and gas outlet is there at the top. But here the main difference is we have a flexible or moving gas holder at the top. So the top of the reactor is not fixed, it is like a floating structure. So that is the main difference here. So floating digester consists of an underground digester, it will be cylindrical or dome shaped, and a moving gas holder. That is the main difference. Here a gas holder is a moving one or a floating one. The gas holder floats directly on the fermentation slurry or on the water jackets of its own. So normally this gas holder floats. In some of the reactors we have some water jackets like an engine where water is filled around this gas holder. So it floats around that water, or in some other case the slurry itself will act as the buoyant force, the necessary buoyant force for this gas holder. And we know when the biogas is produced, automatically the gas holder moves up, and if it is consumed or removed from this through the outlet pipe, it will come down. So that is the movement of gas holder. So when the biogas is produced continuously, the gas holder will move up, and when once it is removed from the gas pipe, it will come down. And it is the gas drum is prevented from tilting by a guiding frame. So you can see a central frame is given in extra to just to prevent it tilting from the help of a guiding frame. And this another important floating drum digester that may widely used in India is the KVIC digester, or Gandhi and Village Industries Commission, Bombay. They have made a slight modifications in this Patel model in 1962, and it is commonly known as the KVIC model. That is another widely used model now in floating type digesters. So you can see here slight differences are there. Here the reactor is fed semi-continuously through an inlet pipe and displays an equal amount of slurry through an outlet pipe. So as the design is such that, suppose if you are feeding a 1 kilogram of biomass through the inlet pipe, it will displace a 1 kilogram of slurry equivalent to 1 kilogram of slurry to the outlet pipe. and the reactor has high height to diameter ratio. So compared to the Jesubhai Patel floating type model, here you can see its height is very high. That height to diameter ratio is very high, and at the central they have a baffle. That baffle helps to prevent the short circuiting of biomass. So what is short circuiting of biomass? Suppose that baffle is not there, there is a chance that some biomass without any anaerobic digestion will go directly to the outlet pipe. So to avoid that, a long height baffle is provided. So it helps to prevent the short circuiting of biomass going out through the outlet pipe. Now these are the two basic designs we have discussed. One is the Patel model, first introduced in India, then a slight modification KVIC model, another floating type digester. Now what are the advantages of floating drum type digester? It's very easy in operation, and gas pressure is constant. That is an important advantage. In fixed dome type, the gas pressure was not constant, it was a varying one. That is a big problem. But here, since our gas holder is a flexible moving one, we will always get or adjust the pressure at a constant level. And another advantage is we can predict this gas volume, how much gas is produced. We don't know anything inside because it is highly insulated from light, sunlight and air, etc. But in this reactor, by looking at the position of drum, so if the position of drum is very high, we can say that there is enough gas production occurring. But if the position of drum is very low in the digester, we can say that it is very low gas production. So like that we can predict the gas volume produced in this type of digester. But the important disadvantage is here the gas holder is mainly made of steel or some metallic structure. So it is very expensive compared to the fixed dome type, where the complete reactor is made of brick, etc. But here the gas holder or even the digester completely itself can made of metals like steel. So it will be expensive and maintenance will be high. And the regular painting should be carried out because the prevent the rusting. So rusting should not be occur in this type of digester. So to prevent that, we have to paint frequently these digesters. And the lifespan also is very short. We have the fixed dome digester, we have seen that is having 20 years of lifespan or more than that. But here maximum 15 years we can predict a lifespan, and mainly if it is installed in coastal regions where there is chance of rusting, etc., this may be reduced to 5 years. Even 5 years the lifespan will be reduced if it is installed in the coastal region. And another problem that may occur is this gas holder may get stuck if our biomass is so highly fibrous structure. Suppose our biomass is some fibrous like structures are there, then this gas holder may stuck, since these fibers will get into the gap between this gas holder and that outside chamber, the water where the water is there. So that is a disadvantage of this type of digesters. So these are a few pictures of, first one is a floating drum type digester installed in Mangalore, and another one is digester installed in Trivandrum, latex, Hindustan latex, utilizing canteen waste. So all you can see is made of metal, and you can see the gas holder at the top, the outlet pipe coming out, etc. Now finally we have a bag type digester, which is also known as the balloon type. So symbol, we have a bag or balloon which act as the biogas digester. So it comprises of a long cylinder, so it will be like an horizontal cylindrical shape. It's made of either PVC or a material known as red mud plastic, which is developed from the residues of bauxite smelted in aluminum production plant. So this balloon digester, they are horizontal in shape and made of PVC or some other plastics like red mud plastics. And here also we have inlet pipe for feeding the biomass and outlet pipe for taking out the slurry, and we have a gas outlet pipe at the top. And this gas produced is stored in the bag itself under a flexible membrane. The feedstock is fed in the bag semi-continuously with the feed replacing an equal amount of slurry. So it will be a not continuously or not batch type, it will be a semi-continuous reactors. So once we are feeding the biomass, it will displace this slurry, equal amount of slurry, like that it will work. And what are the advantage of this? This is very simple and low cost, and the main advantage is transportation is very easy. So we can just purchase a bag and just transport that into any rural location. So installment and construction difficulties are reduced. But the disadvantage is this material, the fabrication material is, if damaged, it will very difficult to repair. So once the bag or this balloon gets damaged, leakage will be there and it will be very difficult to repair this type of plastics. So obviously it is having a relatively short lifespan, not like the other digesters. We have fixed dome digester almost 20 years of lifespan, and the floating drum almost 15 years. But this type of digesters, we cannot expect that type of lifespan. Maybe only 4 or 5 years it will last. And the gas pressure is also very less, since it is only a flexible material of thin plastic. This will be having low gas pressure, and it is also not environmental friendly because these plastics are always considered as a waste once it is use is ended. So it is not an environmental friendly also. So you can see mainly they are applied in Africa because there are rural areas in Africa where this construction and installment of these fixed dome and floating dome digesters are difficult. So they can install these type of bag like digesters. The right one we have seen in Ahmedabad, Gujarat, in India also. And another classification we can say about digesters is according to steps. How many steps or how many reactors are present in the digester? So now so far we have discussed is only of single stage digesters where all the stages of anaerobic digestion is completed in a single tank. So we have only single tank or single reactor where all the stages, what are the 4 stages of digestion, anaerobic digestion: hydrolysis, acidogenesis, acetogenesis and methanogenesis. All these are happening in a single reactor one by one. So that type of reactor is known as a single stage digester. But we can also fabricate multiple stage digesters where the reactors happen in different tank. Suppose we have a 2-stage digester, means what is the meaning? Normally the first 2 steps occur in the one tank and its substrate is transferred into the second tank where the last stage, that is methanogenesis, occurs. So in a 2-stage digester, the first 3 steps or 2 steps are happening in the first reactor or first tank. Then after this the substrate is moved to the second tank where the methanogenesis will occur. So the advantage is this will help to perform the individual bacteria species separately. So we have seen all these 4 stages or 3 stages are done by different types of bacteria. So if they are in a separate tank, they can act fast. And another advantage is we can maintain the proper pH for acidogenic and methanogenic bacteria. If you are happening in a single reactor, we may not be able to maintain the correct pH in all these stages. But if you are having a separate reactor, we can exactly do some and add some enzymes or some acids etc. to maintain the exact pH of that stage. So this is a typical two-stage biogas reactor. You can see first we have a mixing tank. After that the substrate goes to the first stage where hydrolysis and acidogenesis and other stages happen. It is a small reactor. Finally, after that the substrate goes to the second reactor where methanogenesis stage happens. That is the last stage of anaerobic digestion. And the slurry we can recirculate or we can use for other purpose. So this is a block diagram of two-stage digester. And finally we can have another classification that is based on the capacity, that is how much meter cube of this consumption it is having, biomass consumption, how much meter cube it can have. Based on that we can classify into small scale, medium scale and large scale biogas plant. So first one you can see it is a household digester for single house. It is having a capacity 1 to 25 meter cube per day. That much capacity is having. And mainly the small scale digesters we are using the Inbandhu model or KVIC model. So the Inbandhu model is the fixed dome type and KVIC model is the floating dome type. Then in the next level we have some community based biogas plant which is suitable for some canteen, hotels or some flat residential area where a number of houses are there. So in that case we can go for community based biogas plant, a medium scale whose capacity is 20,000 meter cube per day. And the commonly used commercially available digesters in this category are KVIC model, then Bark Nisarguna developed by Bark etc. Then finally we have a large scale biogas plant which is suitable for power production, that is very like in Punjab we have a 1 megawatt anaerobic digestion plant where a large capacity, so it is having capacity of greater than 2500 meter cube per day. And the commonly used techniques are continuously stirred reactor, plug flow reactor, lagoon reactor etc. So these are a new type of reactors based on some principles. So this is an overall classification of anaerobic digestion reactors. So with this we are stopping this session. Thank you.